Everybody, last time we looked at how protein kinase associated receptors can be activated by the growth factors that you find in serum. So what we're going to do in this video is take a look at what happens after you've activated the receptor. And so really we're looking here at the signal transduction process. So I'm going to back up a little bit before receptor activation occurs. And then we'll activate the receptor in a moment. So in a minute, once we get that receptor activated, we're going to see the involvement of some proteins in the cytosol here, these two called GRUB2 and SOS, and then this very, very important protein here called RAS. And so RAS is attached to the cell membrane by either a prenylation or another lipid-like modification. And so RAS is at the membrane there, and then we've got GRUB2 and SOS, which is cytosolic. So once autophosphorylation of the receptor occurs, we're going to see recruitment of these two proteins here. And so this protein here, GRUB2, has a domain that's a region of amino acids that's folded up into a discrete three-dimensional structure. So it's got a domain that recognizes the tyrosines that are going to become phosphorylated when the receptor gets activated. And so GRUB2 has got one of these domains, and it's called a SH2 domain. And that SH2 domain is on the end here, and it's going to recognize phosphorylated tyrosine in the cytosolic tails of the growth factor receptor. So before we activate, GRUB2 won't bind to the receptor, but in a moment we'll activate with the EGF. So the EGF is going to come in and activate the receptor. So there is the EGF binding to the growth factor receptor. The receptors have dimerized. There is our tyrosine kinase domain. And so we've got the trans autophosphorylation has occurred, and we've got these phosphorylated tyrosines in the tails. So let's focus on here over on the right to start with. So what's happened here is that this protein GRUB2 has been recruited to the phosphorylated cytosolic tails of the growth factor receptor. And the point of that the point of binding GRUB2 to the receptor is to get GRUB2 and a protein that it's bound to, SOS, to the cell membrane. So they were kicking around the cytosol. Remember, this protein, just like other proteins we've seen, is going to have an association and a dissociation constant. And so when you get these phosphorylated tyrosines on the tail of the cytosolic tails, is what's basically happening is we're seeing that the association constant, the affinity constant, is becoming much, much tighter, or the binding is becoming much, much tighter. So GRUB2, when it bumps into the receptor now, rather than just bumping in and letting go, it's going to bump and it's going to stay stuck there for much longer. So, and so we've recruited GRUB2 to the receptor along with this important protein here called SOS. So the next thing we're going to look at is what's going on over here with this protein RAS. So RAS is it's often mutated in a number of different types of cancers, and it's very, very important in regulating cellular proliferation. So RAS is a GTPase. Now, we've met GTPases before when we looked at the G protein-coupled receptors, and we looked at G proteins there. Now, those were heterotrimeric G proteins. Remember, they had an alpha, beta, and a gamma subunit. So RAS is a different family of G proteins. It's called a small monomeric G protein. So it's only got one subunit. It's monomeric. And so RAS, like the G proteins involved with the G protein-coupled receptors, RAS binds GDP or GTP, but it's only active when it's bound to GTP. So it's got these two states, active with GTP bound and inactive when it's got GDP bound. So to get into the GTP bound state, RAS has to get some help from this protein SOS. So SOS is one of these guanine nucleotide exchange factors that you've also seen before, which help to switch out the GDP for GTP. So by bringing SOS to the membrane where RAS is located, what we can do is we can interact SOS and RAS, and then we can switch out the GDP from RAS, because if RAS is just in an active state with no receptor activated by ligand, then RAS will just have GDP bound. And so SOS will kick out the GDP, and then RAS will bind GTP, and that turns RAS on. So that activates RAS. And so this whole point of this phosphorylation of these tails is to get SOS to the membrane via GRUB2. So that's this whole pathway over here. We'll come to this protein here, RAF, in a moment. So the other thing that can happen here is that different types of phospholipase C that also have one of these special SH2 domains for binding phosphotyrosine can bind to the cytosolic tails of the receptor. And so when phospholipase C is localized here, remember, it can take PIP3 and it can break that off to form, PIP2, and it can form IP3, which can then go and act on calcium channels, and it can form diacylglycerol, which can then work with calcium to fully activate PKC. So we can also get this receptor working through phospholipase C and some other signaling pathways. So we're going to keep thinking about this pathway over here. And this pathway over here is really the start of what's called the MAP kinase pathway. So once we've got RAS activated, RAS binds to this protein called RAF. RAF gets recruited to RAS when it's GTP bound, and that activates RAF. And RAF is a protein kinase, so that means it can phosphorylate other proteins in the cell, in the cytoplasm. So that initiates this whole phosphorylation cascade. And so RAF, when it gets recruited to RAS, it gets activated, it becomes phosphorylated. It's not phosphorylated by RAS, but it becomes activated by phosphorylation. And then RAF can phosphorylate this protein called MEK. And then MEK, when it becomes active by phosphorylation via RAF, can phosphorylate this protein called MAP kinase, sometimes called ERK. And so MAP kinase gets phosphorylated by MEK, and then MAP kinase is phosphorylated. So we have this pathway, RAS, RAF, MEK, MAP kinase, or as I learned it when I was younger, RAS, RAF, MEK, ERK. And so that's the cascade here. So this is often initiated by a signaling molecule which is stimulating cells to divide. And so those kinds of signaling molecules are called mitogens. So what happens when the MAP kinase is activated? So MAP kinase goes into the nucleus, and then it can phosphorylate a whole bunch of different types of transcription factor, for example, the ETS and the JUN transcription factors. And when these get phosphorylated by MAP kinase, then we end up with activated transcription factors which produce a change in transcription in the cell, and we get a new set of proteins added to the proteome, and the cell is going to have the appropriate response. Now, all the time that that is going on, remember RAS is a GTPase, just like the G proteins in the G protein-coupled receptor cascade that we looked at. And so RAS will hydrolyze the GTP to GDP, and in its GDP-bound form, everything becomes switched off. So RAF won't be recruited and activated. That means that MEK can't be phosphorylated, MAP kinase can't be phosphorylated. These will all get dephosphorylated by a bunch of enzymes called phosphatases, which take the phosphates off, and the whole thing gets switched off. Now, it will stay switched off until another ligand comes around. And so another ligand binds to the receptor, autophosphorylation of the receptor cytosolic tails, recruitment of GRUB2 along with SOS. SOS will activate RAS by kicking out GTP and letting GTP be bound. RAS activates RAF, MEK is activated, kinase is activated, and then we have a transcriptional response. So as long as ligand is present, the signal will be transduced. As soon as the ligand is taken away, then this pathway automatically switches itself off. And that's what you want. You want the default setting here to be off. Because if this is controlling cellular proliferation and the default is on, then you're just going to get cell proliferation all the time. And if you don't want cell proliferation, then you just want this being switched off all the time and turning it on when you need it. It's like lights. Lights have a default, one of those ones with the motion sensors on anyway, they have a default of off. And when you need them, you turn them on. But after a short amount of time, they get turned back off again. That's kind of like how RAS and RAF work here. Okay, so that's how we initiate signal transduction cascade through protein kinase associated RAS receptors. I'll be back soon to talk more about this. See you then.